animal cloning, we uncover the fundamentals of this mysterious but revolutionary science. How does it work? What does it do? Has it already been proved? Get the facts from Professor Riley of the University of Alberta, where she discusses various repercussions of this new science in our modern world. Stay tuned. We will be back after this short break. Cloning is the process by which an entire organism is reproduced from a single cell taken from the parent organism in a genetically identical manner. This means the cloned animal is an exact duplicate in every way of its parent. This includes consisting of the exact same DNA.
There are currently 3 types of cloning that exist in the world: reproductive cloning, therapeutic cloning, or gene cloning. We will only concentrate on reproductive cloning, which is also called somatic cell nuclear transfer, because that is the one that recreates whole organisms. So how it works is that a somatic cell from the original is taken and its nucleus is transferred to an egg cell from which the nucleus has been removed. After being inserted into the egg, the nucleus is reprogrammed by the host cell, and after being stimulated with a shock, it will begin to divide and grow. The egg cell begins to act like a freshly fertilized zygote since it already contains 2 sets of chromosomes and not 1. The egg cell is then implanted into a surrogate mother and carried to term. Dolly was born at the Roslin Institute in Scotland, the only lamb born from 277 attempts to clone the original sheep. They used a reproductive cloning technique, somatic nuclear cell transfer, to produce the clone of a 6-year-old Finn Dorset white sheep. The unfertilized egg cell came from a Scottish blackface sheep. They cultured the embryo for 6 or 7 days to see if it divided and developed normally before implanting it into a surrogate mother, another Scottish blackface sheep. From 277 cell fusions, 29 early embryos developed, but only 13 were viable enough to be implanted into surrogate mothers. Only 1 pregnancy went to full term, and the 6.6 kilogram Finn Dorset lamb was born after 148 days. Dolly mated and produced offspring normally, showing that cloned animals can reproduce. She was born on July 5, 1996, but was euthanized on February 14, 2003, when she was aged 6.5, about half of the average lifetime of a sheep.
Among this famous example, there are many other types of animals who have been successfully cloned, such as fish, mice, cows, goats, pigs, cats, mules, dogs, horses, monkeys, and a few other types.
I'm here today at the University of Alberta with Professor June Riley, geneticist at the university, to discuss how animal cloning could benefit society, but also the negative impacts it could provoke. Ms. Riley, what would you say are some of the aspects in which animal cloning is a benefit?
Some benefits of animal cloning are basically, first of all, as an example, cloned sheep, they have been genetically modified to produce milk that contains a human protein essential for blood clotting, and the goal is that someday this protein can be purified from the milk and given to humans whose blood does not clot properly. Another example is that cloned animals, they're all genetically identical, and their response to drugs during drug testing should always be the same, which makes for more constant results during drug testing, which is very useful. The FDA also approved that the meat and milk from cloned animals was just as safe and healthy as those from non-cloned animals, and we could also rebuild populations of endangered or extinct species. And on a more familiar side, even though it's costly, grieving families can be given back their pet as long as they have their DNA if the pet is deceased or about to die.
And Professor, what would you say would be some of the negative aspects of animal cloning?
Well, first of all, cloning is a very inefficient and expensive technique since many of the cloned embryos die early or do not develop into healthy individuals. For example, Dolly was the only viable embryo out of 277 other ones. Also, some problems have been noted in the cloned individual, like increase in birth size, a variety of defects in vital organs, like the liver, the brain, the heart, there's premature aging, and problems with the immune system. One aspect that is constantly a problem is the fact that chromosomes have telomeres, which are the tips of the chromosome, and they shrink through each cell division during a lifetime. Over time, the telomeres become too short, making the cell unable to divide, and eventually it leads to the cell's death, which is the normal process of aging in life. And as such, an older embryo is used to clone an animal, and its chromosomes are already shorter than normal. This reduces the lifespan of the clone, since it's genetically older than it appears to be phenotypically. Like, for example, Dolly was cloned from a 6-year-old embryo, died after only 6 years, which is half of the life of a regular sheep, 12 years. And, of course, on the ethical side, cloning goes against, like, religious and societal values about human dignity, going against the ideas of, like, individuality, freedom, identity, and, like, we could produce a clone of a person that once existed or still exists, and that would stir up a lot of conflict and ethical, religious beliefs, and all sorts of things like that.
Thank you, Professor Riley. And that's all for today on the Daily Gene.